I'm Catherine Hitchcock, and I'm a senior scientist in the Forensic Biology DNA Laboratory at New South Wales Health Pathology Forensic and Analytical Science Service, or FASS. And I'm thrilled to chair this plenary session today. Our speaker is a colleague of mine, also from FASS. I hope everyone is excited as me to hear about various DNA tools that assist with DNA identifications, which give hope to victims of crime and families of missing persons. I'm very pleased to introduce Carole Field, who will be presenting DNA-led identification for criminal and coronial casework. Over to you, Carole.
Thank you, Catherine. I will just present my screen. And again, thank you, Catherine, for that lovely introduction. I am very excited today to talk to you about DNA-led identification for criminal and coronial casework. So the Forensic Biology Laboratory is the primary provider for any criminal or coronial casework, for DNA testing for criminal and coronial casework within New South Wales. We're basically 3 different units, but there is overlap, and there are many staff that are cross-skilled so that they can work in multiple units. Our evidence recovery unit is where examination of crime scene exhibits is carried out. Now, the majority of samples that they receive in the evidence recovery unit are sexual assault investigation kits, but they also receive ante-mortem samples from missing persons cases and post-mortem samples. Now, the post-mortem samples can be for identification purposes, or it might be a known victim of a homicide where we are looking for foreign DNA. Most of the exhibits from crime scenes are actually examined by the New South Wales Police, and they come into the laboratory in specialised tubes that can just go straight onto our robots. So they'll bypass our evidence recovery unit and go straight to the DNA processing unit. The DNA unit is also responsible for testing the reference samples from persons, such as convicted offenders, suspects, victims, and of course, the relatives of missing persons. When the DNA testing has been completed, the results are available for the case management unit to interpret and report. Now, we are all electronic, so the results are all reported to New South Wales Police electronically. The case management unit is also responsible for maintaining the DNA database, where the DNA profiles are uploaded to the DNA database for searching purposes, and any links are provided to New South Wales Police. The case management unit also provide investigation reports and court statements, and are also available for expert testimony for court. So routinely in our laboratory, we are carrying out 3 different types of DNA testing: autosomal DNA, Y chromosome DNA, and mitochondrial DNA. Now, the autosomal and the Y chromosome DNA are contained within the nucleus of the cell, and they're often referred to as nuclear DNA. The mitochondrial DNA is a completely different type of DNA, and it's contained within the mitochondria of a cell. Most of the DNA from individuals is exactly the same. That's what makes us human. There's only really a very small percentage of DNA, about 0.1%, that actually is different between individuals. And it's these variable regions that we target in forensics to enable us to determine the difference between one person's profile and another. The exception, of course, is identical twins. They do have the same DNA. So if you're a twin and you're going to commit crime, you could potentially get away with it by blaming it on your other twin. That is, of course, as long as they don't have a really good alibi. Now, most of the DNA is found in almost every cell of the body. The exception is red blood cells. But it doesn't matter where you actually take a sample from, whether it's hair, bone, semen, saliva, or blood, the DNA profile will be exactly the same. And biological fluids are a very rich source of DNA, so we don't need much to get a DNA profile. So what do we use DNA for? We are only interested in identification. We are not looking at any health-related information. In fact, almost all of the DNA testing that we currently do in the laboratory does not contain health-related data. We are purely interested in that identification. Now, there are a number of processes or steps involved in obtaining a DNA profile. Now, by far, the most common test carried out in the laboratory is for autosomal DNA. So it starts off with the sample being placed into a specialized tube that can go onto a robot. Now, this can either happen by New South Wales Police at their crime scenes units, or it will be our scientists in our evidence recovery unit. Now, the first step is to break open the cells so that you can, release the DNA. We then need to separate the DNA or extract it from the other cellular material. And then it's important to find out how much DNA is in that sample. Now, that's important before the next step in the process. Too much DNA can cause some issues, but also too little DNA may mean that you end up with a very partial DNA profile. The next step will involve copying the DNA fragments over and over again, a bit like a molecular photocopier. And the last step is where we run it through our instrumentation, our capillary electrophoresis, and this just allows us to be able to visualize that DNA profile at the end. So just a picture of some of our robots. On the left, we have our lysis and extraction robots, where the sample will be purified. In the middle, we have where we are quantifying, so finding out how much DNA is in the sample and also copying it millions and millions of times so that we can then visualize the profile through our analyzers that are here on the right. And these just allow us to actually visualize that DNA profile. The end result is an electropherogram, which is just a graphical representation of a DNA profile. So now I'd like to talk a bit about missing persons casework. Now, the New South Wales Police are responsible for sending the exhibits to our laboratory for testing. And this includes unidentified human remains, but also reference samples from missing persons and their relatives. So why do we use DNA to identify human remains? DNA is a very powerful tool that can distinguish biological samples originating from different individuals. It remains constant through our life. And as I mentioned, it doesn't matter where you sample the DNA, whether it's from tissue, blood, bone, that DNA profile will be the same. DNA is inherited from our parents, and that's actually really important for identification of missing persons. DNA can also be recovered from old and degraded biological samples. And if the human remains are fragmented, then DNA may be the only form of identification. So once you have your DNA profile, you really need something to compare it to. And the best sample for comparison is an actual sample from the missing person. And we call these direct matching antemortem samples. The ideal sample is a medical sample or a newborn screening card from the missing person. Unfortunately, these are often not available. So the next option is to actually use personal effects from the missing person, such as a toothbrush or a hairbrush, a razor, that sort of thing. Now, although direct matching antemortem samples is the best form of identification, we do sometimes have problems with personal effects. It's not uncommon to obtain a mixed DNA profile. And by mixed, I mean there's more than one person on that DNA profile. And you can even get that from toothbrushes. Now, I'm not saying people are sharing toothbrushes. They may just share a bathroom. So if only personal effects are available, we ask police to bring in multiple samples, but also to bring in reference samples from close family members, because this way we can compare the profile from the toothbrush to the family member and just ensure that there is that biological relationship. Now, if there are no direct matching antemortem samples, then DNA identification will depend on having reference samples from your family members. And a reference sample is just a known sample from a person. And it's generally taken by using a swab and just swabbing the inside of your cheek. For missing persons casework, the samples from the relatives of missing persons are only allowed to be searched on the database against unidentified remains. A few years ago, we increased our focus on missing persons casework. And this was done through a project called the Human Skeletal Remains Initiative. We worked in collaboration with the mortuary and the New South Wales Police to conduct an audit on all unidentified remains in New South Wales. Now, this included a review of the historical skeletal remains held at the mortuary, those that had not previously been sampled for DNA. Now, some of these skeletal remains were from autopsies that had been conducted a long time before DNA testing was available in New South Wales. The New South Wales Police, they reviewed their long-term missing persons cases, and they found that many of those didn't have that direct matching antemortem sample. So they put a focus on obtaining family member profiles from the long-term missing persons cases. Now, our part in this initiative was to review our holdings of unidentified remains and the reference samples from the relatives. And we found that many of those had actually been typed in an older DNA typing kit that doesn't give you as much information. So they were retested with the latest autosomal DNA typing kit that we're using. We also did Y chromosome testing in all the male unidentified remains and all the paternal relatives, and mitochondrial DNA testing on the unknown remains and the maternal relatives of missing persons. So lots of further testing involved, but we were gearing up for familial searching. And having the multiple different types of DNA testing has greatly increased our capacity for searching. The New South Wales Police Missing Persons Registry introduced pop-up DNA collection centres in regional areas around New South Wales. So these centres gave the relatives of missing persons a place to come along, give some information about their missing relative, but more importantly for us is that enable them to give a DNA reference sample that could be used for familial searching. And I'm just going to talk briefly about the different types of DNA testing that we carry out. As I said, by far the most common test carried out in the laboratory is autosomal DNA. It is inherited from our parents. We receive half from our father and half from our mother. So autosomal DNA has the greatest power of discrimination. So that's generally where we go to first. Now, as I mentioned, if there are no direct matching antemortem samples for a missing person, then the DNA identification is going to rely on having reference samples from their relatives. So what we do is we ask police to, where possible, can you give us a mother, father, child trio? So if the child is missing, a profile from the mother and the father is ideal, because this way we'll get a complete picture of this missing person. If it's the father or the mother that's missing, then we ask police to bring in profile from the child or children of the missing person, but also bring in the partner, because that way we can still get our mother, father, child trio. But we can search complex pedigrees. So if that trio is not available, we just ask police to make sure that multiple relatives are tested. And the autosomal DNA is searching nationally. So our current autosomal DNA typing kit is called PowerPlex 21. You might see that on the slides as PP21 also. So we've been using this typing kit since 2012. Prior to that time, we were using a different typing kit called Profiler Plus that didn't quite look at as much DNA, anywhere near as much DNA as our PowerPlex 21 kit. PowerPlex 21 targets 21 areas of the DNA. One of those is a sex marker. Females have the X chromosomes and males have an X and a Y. So as I said, a female only has X chromosomes. she has two. One she's received from her father, one she's received from her mother. So because she only has X chromosomes, she will pass an X onto her sons and her daughters. Now, because she's got two X's, the DNA information can actually get mixed up between them. So what she's passing on could be whatever she's received from her parent or a mixture of both. The male determines the sex of a child. So if he passes on an X, the child will be female. If he passes on a Y, the child will be male. No, he doesn't have two Ys. So the information on that Y chromosome can't get mixed up. That Y chromosome is passed on from father to son intact. And that's really important for the work we're doing in the laboratory. Now, this is just a picture of part of an electropharogram for a Y profile. But as I said, that unique, the fact that Y is passed on from father to son without getting mixed up is really important. And it's actually very, very important in sexual assault cases. And I'll just sort of go off on a tangent here for a moment. In most sexual assault investigations, you start looking for sperm. And if you find sperm, that's fantastic. We can separate the sperm cells from the other cellular material, and you'll get your DNA profile from the sperm. But what if there, is no sperm? Maybe there's no ejaculation. Maybe the offender's had a vasectomy or the condom being used. When we're investigating intimate swabs from a female sexual assault victim, and I will say, but the majority of sexual assaults are a female victim and a male offender. So when we're looking at those intimate swabs from the female victim, she's going to have bucket loads of DNA in that sample. And her DNA can completely swamp any foreign DNA. So trying to find a small amount of male DNA in amongst lots and lots of female DNA is not going to work with our routine autosomal testing. That's where the Y testing comes in. That Y is specific for only targeting the Y chromosome that's only found in males. So this way we can find a small amount of male DNA in amongst a large amount of female DNA. And I'll talk more about the Y chromosome searching later. So for missing persons casework, we actually use the Y a little bit differently. Because it's only in males and because that Y is passed on from father to son intact, it actually means that all males on that same paternal line will have the same Y DNA profile. So where a first degree relative is not available for a missing person investigation, any male on that paternal line can be used for identification purposes. And the Y chromosomes are searching nationally. Mitochondrial DNA also has a unique inheritance pattern. Mitochondrial DNA is passed on from mother to children, both her sons and her daughters, and then her daughters will pass it on down the maternal line. So again, all maternal relatives will have the same mitochondrial DNA, which is really useful in a missing persons case because any of those maternal relatives can be used for comparison. Mitochondrial DNA is also really useful for highly degraded samples, which often happens for unidentified remains. And again, the mitochondrial DNA profiles are searching nationally. So in New South Wales, we have a state and a national database, and the autosomal DNA profiles from the unidentified remains are uploaded to both. Now, if you are dealing with historic human remains, when you upload that DNA profile, that autosomal DNA profile onto the database, if you don't get a direct match immediately, well, you're not going to get one in the future. So the only options you have is familial searching using autosomal DNA or searching with a lineage marker, such as mitochondrial or Y. So what is a familial search? It's one of the tools in our DNA toolbox. It's an intelligence tool that may give you an investigative lead. It involves a trawl through a DNA database, looking for individuals that share DNA with that source or that searching profile. No, as I said, the search is performed with autosomal DNA. And what it will do is give you a list of potential relatives that will need further investigation. Now, it's often quite labour intensive, and that's partly to do with the fact that the composition of our database still has a lot of those profiles that were tested in only an older DNA typing kit. So depending on the composition of the database will depend on how easy it is to actually obtain a relative in your familial search. Of course, so when you run the search, you'll often find those candidates that need to be upgraded. So we do the upgrading from the old typing kit to the new typing kit. And we also will look at any male on the candidate list. If the unknown remain is male, then all males on the candidate list will need to have Y-DNA testing. So that further testing is all carried out. When that's completed, the search is run again. So it can be quite labour intensive. And we have done hundreds of these familial searches. We do find that nearly always, when you've done all that further testing, they're all eliminated as being a relative. So you're left with nothing at the end. But it's really important to do that work because we want to identify these unidentified remains, but we also are increasing the power of our database for every sample that we upgrade from our older kit to our newer kit. And for every Y profile we put on the database, that's adding power to the Y database. So familial searching is really good for detecting first-degree relatives. And that's here, your parents, children, and your siblings. But we can, and we do, pick up relatives outside of first degree. And it just depends on how much DNA they share. And I've got some examples to give later. So familial searching is carried out for coronial and criminal casework. Now, for coronial casework, there is no authorization required. We do the testing routinely. And we use a low threshold to search, and I'll talk more about that in a moment. For criminal casework, familial searching is strictly controlled by the New South Wales Police. The crime must be of a serious nature or a sexual assault, and it requires authorization from an Assistant Commissioner of New South Wales Police. The search is also limited to only searching convicted offenders and suspects on the DNA database. And we will do high and low threshold searches depending on the request from New South Wales Police. Now, the software will rank the profiles when it compares the whole database. It ranks the profiles according to the statistical likelihood for a specific biological relationship, whether that's a parent-child relationship or a sibling relationship. But we all share DNA. On average, related individuals will share more DNA than unrelated individuals. But, you know, that's an average, and we know what averages are like. So a low threshold search is really going to maximize your ability to detect a true relative. But what you find is you're in that area where it's a bit difficult to determine whether someone's related or not. So you do get adventitious matches, and you will need to investigate and do further testing on those adventitious matches. The high threshold search is really like getting low-hanging fruit. You're going to reduce the number of adventitious matches that you get, but you can sometimes miss a true relative. So it's a bit of a balancing action. There are limitations, of course, to familial searching. The power of the search is really dependent on the database size and the composition, as I mentioned. So the more people on a database, the more likely you are to get a link. And the composition is really that balance of how many are typed in the new kit as opposed to the old kit, and how many partial DNA profiles do you have on there. You do have the relative of the searching profile must obviously be on the database to actually obtain that link, but there is that increased probability of finding them if they are typed in our latest typing kit called PowerPlex 21. Now, a parent-child relationship is really the best thing for familial searching, and it's because they've got that defined inheritance pattern. We know they're going to share 50% of their DNA. We know when we look at 20 areas of the DNA, they must share one DNA type at all of those 20 areas. Siblings range in the amount of DNA they share. Siblings still get 50% of their DNA from their father and 50% from their mother, but they can get different bits of DNA. So some siblings can share a lot and some not much at all, and anything in between. So familial searching for unidentified remains and missing person cases has been routinely carried out in our laboratory since 2019. At the moment, we have about 126 unidentified remains on the national database. Now, when we get a new unidentified remain, it is the autosomal profiles uploaded onto the database. If we don't get that direct match, then it will follow the familial searching workflow. The first search that they will have is against the New South Wales relatives of missing persons profiles. So that's what we want to link to. But if there is no match there, then that search is expanded to include the convicted offenders and suspects and the unlimited volunteer samples on the New South Wales database. If there's no match in New South Wales, then the sample will still be stay on the national database searching. Now, we have over 1,000 profiles from the relatives of missing persons. Now, when we get a new one, they will be first initially searched against the unidentified remains in New South Wales. Now, as I have mentioned, the profiles from the relatives of missing persons are only allowed to search against unidentified remain profiles. So if there's no match in New South Wales, the sample is put onto the national database for familial searching. Now, the component of the national database that we use is, we call it NIFA, and that's just where it has the specialised searching that we can do. And if there are multiple relatives for a missing person, we actually create a little family tree for them, and we search with the family tree because that's the most powerful search for a missing person. So if we have mum and dad's profile for a missing child, mum and dad is built into that family tree, and we're searching with mum and dad's profile. So now I just want to talk about some case studies. They're usually the most interesting part of a talk, I think. Now this one, a colleague of mine, Alice, presented this morning about familial searching for criminal matters. And she presented the results of a murder case from 2008 that was solved with a familial search. Now, I will say, when we gave that familial search report to the New South Wales Police, that was an easy solve for them because that one had a parent-child relationship. And as I said, parent-child relationships are the best for familial searching. The police look at that result. They'll look at the date the offence happened. They'll look at the date of birth of the person we are getting a familial link to. And that will tell them whether they're going to look for the father or the child. I say father because most males on the database, most persons on the database are male, and most offenders are male, unfortunately. I don't know whether they get caught more often or they just commit more crime. I'm not sure about that. So that 2008 murder was easy to solve, but not every familial search is easy to solve. And I wanted to talk today about our first ever criminal familial hit. This happened quite some time ago. Now, there was a string of sexual assault incidents, and they happened from the late 80s all the way through to the early 2000s. There was lots of DNA cases involved here. Now, there was semen in all of these cases. So semen's a biological fluid, easy to get a DNA profile from. So the DNA profile was uploaded to the database, and all of these different crime scenes were linking together. But there was no link to a person sample on the database. So no link to a reference sample of a person. Now, over the years, you know, it stays, once the sample's uploaded to the database, it stays on there. So it's continuously searching. This case was one of our pilot cases for familial searching in 2013. So it had its first familial search back then. Low threshold search, we wanted to maximize the ability of finding someone. And when we run the search, we had about 30 odd people on the candidate list. And that was quite low because back in 2013, the majority of our database consisted of profiles tested in an older DNA typing kit, which had limited information. So there was about 30 odd profiles. We had to upgrade them to our latest autosomal kit. All the males had to have a Y-DNA test carried out. All that further testing was carried out. Run the test again. Everybody was excluded. Nothing, no match. No, in 2013, the familial search was a point-in-time search. More recently, familial searching occurs every evening. But back then, we had to repeat the search periodically. So in 2016, when we repeated this search, we again got some people on the candidate list, some males, did the Y testing, and one of those males came back with a matching Y profile. Now, what that's meaning is you've got an autosomal familial link and a matching Y profile. That's actually indicating that you may have linked to a paternal relative of the donor of that crime scene profile. So we were really excited here, but it wasn't a parent-child situation, so it wasn't going to be that easy to solve. And the statistic for that sibling relationship was a bit slow, bit low. So, it was in that range where, you can't tell whether people are related or not, but that matching Y was the difference. That matching Y was pointing us to a paternal relative. So the information was provided to police. Our thoughts were that maybe it wasn't a first-degree relative, so which does make it harder to solve. The police were really excited to go to investigate this one, and we started getting covert samples into the laboratory. Now, I don't know whether you know what a covert sample is, but if police have, for a very serious crime, like a sexual assault, if police have a person of interest, then they can get authorization to follow that person around, and anything they throw away can be used to get their DNA profile. So cigarettes, coffee cups, drink bottles, they're perfect for getting DNA profiles from. So the police would do this. They were investigating this family now. They knew it's not this person, right? It's not the person we linked in 2016, but it's potentially a paternal relative of theirs. So they start building a family tree. They start looking into this family. But we were sending in covert samples, we were comparing them, and they were all excluded. We were not getting anywhere. Then in 2020, we started Y-DNA searching, and we got a Y-DNA search. Now, this was to a different individual, right? And it was a different surname. Now, when you think of that Y-DNA being passed from father to son to father to son, technically it should be the same surname, but maybe this was, 10 generations back. Maybe there was some misattributed parentage, or maybe dad wasn't around and the child took the mother's maiden name. There's lots of different reasons why it could be a different surname, but we gave that information to the police, and they started building up the family trees from both of these individuals and trying to find a connection. This was 2020. That was that wonderful year of lockdowns. Not a good time to do covert sampling. So it took police quite some time before they were able to get some covert samples to send into the laboratory. And it was only this year they took a sample from someone who they thought could be the son of the offender. And guess what? We got that parent-child situation now. So as soon as we told police that, very soon afterwards, we had a reference sample sent into the laboratory that matched the crime scene samples. So this case, although the first, incident of this sexual assault, the first offence was back in the late 80s. It took till this year, over 30 years to solve this case, and really over 6 years since we first got the familial hit. So not all cases are easy to solve, but we keep at it. And that's the important thing. So with that Y-DNA testing, that is actually extending our reach on a familial search, because any male in that paternal line can link on the Y. And that Y link then gets our attention. Now, this one is another serial sexual assault case. There was over 13 sexual assaults from inner Sydney back in the early 2000s. And they were linked by MO and location, and some of them were linked by DNA. No, there was semen in these cases too. So the beautiful semen profile uploaded onto the database, but no person links. The scenes would link together, but no link to a person. Now, there was exhaustive investigations carried out in this case, and that included some assistance from the FBI and the investigation of over 3,000 persons of interest. This was also one of the pilot cases for familial searching back in 2013. But unfortunately, the familial searching didn't give us any strong candidates. It was repeated over the years, still no strong candidates. Then along comes Y-DNA searching, and we get a Y-DNA link. So when we get a Y-DNA link, we'll still look at the autosomal profile. For this person, there was no support at all, or no statistical support for a close family relationship. So I know we can't ever completely exclude a sibling with autosomal DNA, but it was more likely it was going to be further than a first-degree relative. So we give this information to the police, and they start their investigation. Now, when they're investigating, they're going to look into this family. They're going to look at other males within this paternal line, and to see if they can find somebody that potentially could fit into this crime. And they found someone, and he ended up being a half cousin of the person that we linked to with the Y-DNA link. A half cousin will not share enough DNA to appear on our candidate list in a familial search. They're just too distant. But the power of that Y-DNA and that extended reach of that Y-DNA brought this person to our attention, and therefore the attention of New South Wales Police. A reference sample was brought in from that person of interest, and it matched to the crime scene samples. And he was arrested and charged. And this is another one of our Y-DNA cases, and you might know where I'm going here with the title, Bad Brother. This was a sexual assault that was carried out in 2014. The complainant awoke in a police car with no memory of the assault or the description of the offender. Now, I have to qualify here, the sexual assault did not occur in the police car. The complainant had passed out and the police were assisting. So there was semen in this case. So we had a beautiful autosomal profile. We had a Y profile uploaded. Well, the autosomal profiles uploaded onto the database. This is 2014. No Y searching in 2014, but we had a Y profile developed because we were doing familial searching. So there was no match on the database for the PP21 profile, for that autosomal profile, and no strong candidates in our familial search. And again, the familial searches are repeated periodically, but again, no strong candidate. Until we start our Y-DNA searching, and we got a Y-DNA link to a person. And when the police investigated this one, they realized that he was the brother of the person that we'd linked to. So this is a case where there's a small proportion of siblings that just don't share enough DNA to appear on our candidate list for a familial search. But that Y-DNA, it's got that reach. It can find them for us. Now I just got some missing person casework I'd like to talk to you about. This one's a bit unusual in it's an historic case, but sometimes they're difficult to identify. So in 1997, the body of a badly decomposed male was recovered in Sydney Water by Sydney Water Police. In 2001, a blood sample was sent by New South Wales Police to another laboratory for mitochondrial DNA testing. Now, back in 2001, we were just starting autosomal testing. We could not do mitochondrial DNA testing at that stage. Through the Human Skeletal Remains Initiative in 2018, we were actually provided with the mitochondrial DNA testing report for this case. Just the report. We didn't actually have a sample to test ourselves. So, again, through the Human Skeletal Remains Initiative, mitochondrial database searching started. Unfortunately, for this case, it had a common mitochondrial profile. And by that, I mean, it just by chance could link up to other individuals that are not on the same maternal line. And that's what happened in this case. This mitochondrial profile matched 3 relatives of missing persons, so 3 different missing persons. One of them was female, so they were easy to eliminate, but we were left with 2 males, that this unidentified remain could potentially be one of these 2 males. Now, more DNA information is always better. Give me more DNA. I can do so much more with more DNA. But all we had was a mitochondrial profile in this case, and a common one at that. But even though this person died in 1997, through communication with the New South Wales Police and the mortuary, we were able to source a sample from this individual, this unidentified remain. So we did our usual testing, our autosomal testing, our Y chromosome testing, and then compared those to the relatives from those 2 missing persons. Unfortunately, they were both excluded with the autosomal DNA. We did our usual upload to the database, familial searching, no match. We were back to the drawing board with this one, but at least we had more DNA information. Now, the New South Wales Police started new investigations with this, and they were able to provide us with a reference sample from the mother of a missing person. And this was she was from the UK. Now, unfortunately, there can be problems when you're comparing DNA profiles between countries, because we're usually using different DNA typing kits. And sometimes there's limited overlap. Unfortunately, there was limited overlap here. What we could see, there was a potential parent-child match, but it was limited DNA. And I can't think of anything worse than telling someone, we have phoned your son, to find out through further testing that it's not. The mother of this missed person was quite elderly, and they were in the UK. She had not had mitochondrial testing carried out on her sample. So that was going to be a little bit hard to organise. But we had a Y profile in this case. So, I was asking the police, can you get us a paternal relative, any paternal relative? Fortunately, the father of the missing person was deceased. But the investigator was looking, there was a potential paternal relative over in the US, so they could look into that. But as luck would have it, the father of this missing person, although he was deceased, he died in New South Wales. So we were able to get a stored sample from him. And that gave us that beautiful mother-father-child trio with a lovely identification, a very confident identification. Now, this one's an old case. It's a very interesting case, and it involves multiple jurisdictions. And I wanted to talk about this one because it also happened, even though it's an old case, it happened before we could do that national familial searching. So I just wanted to contrast what we would do differently today. And I'm sure you're going to recognise this one. 2010, human remains were recovered in Belanglo State Forest. Now, I know you're all thinking of Ivan Milat now, but it was not him. The remains were from a female individual between 13 years and 25 years. And it was estimated that her bones had been there from anything from 6 months to 10 years. So not a lot to go on. But we got a beautiful autosomal DNA profile from it, uploaded onto the database for direct matching, but no match. No, there was a media release for this. It included the possible image of this unidentified female, and this was created through the work of a facial anthropologist. And the media release also included a t-shirt, an image of a t-shirt that had been found near her bones. So she was known as the Angel of Belanglo for quite some time, and it was as a result of this T-shirt. But of course, the media releases are done in the hope that people will recognise her. But unfortunately, that was not the case for here. She remained unidentified for a number of years. Then in 2015, the skeletal remains of a child were discovered in a suitcase on the highway in South Australia. Now, there was clothing and other items in the suitcase. Images of the clothing and a blanket from the handmade blanket from the suitcase was actually, sent out to the media, sent out by media. And again, this is so the public can have a look and hopefully recognise them. And these images were actually pivotal to this investigation. There was a call to Crime Stoppers. The person that called was able to provide the police with a photograph of a child wearing the exact clothing from the suitcase. She was the friend of the mother of this child. This gave South Australian Police a lead to follow, and they ran with it. It was thought that this person, the child in the suitcase, was Candice Pearce-Stevenson. Now, she had lived with her mother in Alice Springs in the Northern Territory. So a Guthrie card, or a newborn screening card, was obtained for Candice, and that confirmed that she was in fact the child inside the suitcase. So now the investigation turned to, well, where was Candice's mother? Now, New South Wales Police were also in touch with South Australian Police, and they knew about our Belanglo girl. So they sent us a DNA profile from Candice so that we could compare it to the DNA profile we had recovered from the bones from Belanglo. And they were a mother-daughter match. So very soon then, the angel of Belanglo was confirmed to be Carole Pearce-Stevenson, using a Guthrie card from her. So what would be different today? What would have happened if there was no clothing in that suitcase? Would Carole and Candice still be unidentified? What we can do now is we have the ability to do familial searching at a national level. So a familial link would have been obtained from the unidentified remains in New South Wales and the unidentified remains in South Australia. And just to finish this case study, Daniel Holdham, who was Carly's boyfriend at the time, a lot older than Carole and not a very nice person, he became the suspect in the murder case. The partners are always the suspects, but he was eventually charged and convicted and sentenced for the murders of Candice and Carole. Now, last case study, this is our lost mariner. Back in 24th of September 2011, a jawbone washed up on the New South Wales beach. Now, a male DNA profile was recovered from this jawbone, and the autosomal profile was uploaded onto the database, and there was no match. That seems to be a common occurrence, doesn't it? Now, back in 2011, there was no familial searching. So over the years, the New South Wales Police would call us up and ask us to manually compare the DNA profiles between the jawbone and nominated reference samples from relatives of missing persons. But we couldn't identify them. It wasn't till 2020 when a familial search produced a link on the New South Wales database. And this was to a male candidate. So Y-DNA testing was carried out, and he came back with a matching Y-DNA profile. So what have we got? We've got an autosomal link and a matching Y profile indicating this is potentially a paternal relative here. So we gave that information to the police. When they interviewed the candidate from this familial search, he told them that his Uncle Bill had been lost at sea in a boating tragedy. Now, this had happened a long time before he was born. He had very little information about it, but that gave the police a lead. Bill Moran went missing in 1979. Now, that's over 40 years ago. He was on a small cabin cruiser with his wife, Philippa, and 3 others. The seas became treacherous, the weather changed, eventually, you know, they battled these enormous waves for so long, but eventually their vessel was sunk. And they were left with an aluminium dinghy. But the huge waves capsized the dinghy also. Now, Bill and his wife managed to stay with the dinghy, but the other three were washed away. The ship's radio had actually been damaged early in the ordeal, but they continued to send mayday calls. They didn't know if anybody could hear them, but someone did. The three people that had been washed away from that dinghy were actually rescued by helicopter after 6 hours in the water. 2 days later, the body of Bill's wife, Philippa, was recovered on a beach with some wreckage from the cruiser. Bill's body was not recovered in 1979. It was assumed that Bill had drowned when his vessel sunk on the 24th of September, 1979, but his jawbone would turn up on that beach exactly 32 years later. Now, Bill's brother, he's still alive today, but we didn't have his DNA reference sample. Bill went missing in 1979. We weren't even thinking about DNA testing back in 1979. But through Bill's nephew and a familial search, we were able to provide some closure to the Moran family after decades of searching for answers. And if you'd like to know more about this one, it's very interesting. The New South Wales Police have the State Crime Command have a podcast called Lost at Sea. It's a very heroic sea rescue. So just to finish up, what else do we have in our DNA toolbox? Well, if you have no direct database match and there is no familial match, there is still a way that you can get an investigative lead through DNA. Specialized DNA testing can be carried out to identify the eye color and hair color and skin color of an unidentified profile. We can also get biogeographical ancestry information, so where their ancestors have come from. Now, in a missing persons case, missing person casework, this could narrow down the number of missing persons that this could potentially be. Or if it's a criminal investigation, if there are no suspects, this can provide a lead. Or if there are lots of suspects, it may help to narrow down that suspect pool. So hopefully I have shown you that familial searching is a really good investigative tool that can assist in the identification of human remains and criminal casework. Now, there is a new tool though in the DNA toolbox, and it is called forensic genetic genealogy. Now, it uses different DNA typing than we currently do in our laboratory. It also uses different databases. Now, if we've got doing our familial search, we're using a government DNA database. Forensic genetic genealogy is using a public database. I like to think of this as familial searching on steroids. And that's because they look at a lot more of the DNA than we can currently do. So not only are you finding first-degree relatives, you're getting the grandparents, the grandchildren, the aunts, uncles, nieces, nephews, and cousins. And not just first cousins, you're getting second cousins and third cousins and more distant cousins. Now, this is a picture of Marsha King. Her body was discovered in 1981 in the US. She had been strangled and beaten. She was known as the Buckskin Girl for many years, until she was identified in 2018 through forensic genetic genealogy. And since that time, hundreds of violent criminal cases and John and Jane Doe cases have been identified in the US. And this is coming soon to New South Wales. Watch this space. Thank you.
Thank you, Carole, for that amazing presentation. That was fantastic. So now I'm going to open the floor for questions if anybody has any. Actually, I can see.
I'll just stop sharing.
Sure. I can see some in the question box. The question is, do you choose what cases can get familial searched, or is the request made by police?
It actually can come from both. It has to be authorised for a criminal case, that is, because we can do all the searching we want for unidentified remains. For a criminal case, it has to be authorised by New South Wales Police. But we can, in the course of our casework, we may come across a serious crime or a sexual assault case that we believe would be a great candidate for familial searching. We can tell police about that. They just get the authorisation. So it works both ways.
Mm-hmm. And you mentioned that familial searching is applied to serious crime cases. Do you imagine that it will be used in the future on less serious crimes, so that might disrupt the those kind of less serious cases like break and enters, for example?
Well, again, it's ultimately the decision is with New South Wales Police. It is they have to authorise the search. But I will point out, those less serious cases like break and enters and, stolen motor vehicles, they actually impact the public more than a serious crime case does. If you think about it, if you talk to someone, they may have had that, break and enter happen to them. So the impact to the society can be big for those, what we call volume or property crime cases. If there is a string of property crime cases, then the police will probably look at that. And if there's multiple cases, if there's continual cases linking, so that you've got lots of DNA cases linking on the database, then they may look at that and decide to actually authorise a familial search. So again, it's all with the New South Wales Police.
Okay. Well, thanks again. Thanks very much for that presentation. I think we've come to the end of the session now. So I just have to remind people to make sure you take a look at all the posters during the break, ask any questions of our poster presenters, and don't forget to click on the link to join the Early Career Researcher session, which will be after this break. And the break goes until 10:55. So we'll see you soon. Thanks again,
Thank you, Catherine.
Carole. Thanks.